Hi guys, welcome to this YouTube channel. A good day to everyone watching this video. So in this video, I am going to primarily discuss the answer key for the GATE Excel Biochemistry previous year's questions of the 2023 paper. So let's get started. So the first question, determine the correctness or otherwise of the following assertion A and the reason R. So the assertion is on a per carbon basis, palmitic acid yields more ATP than glucose. The reason is carbons in palmitic acid are more reduced than those in glucose. So the correct answer here is option A. That is, both assertion and reason are true, and the reason is the correct reason for the assertion. So the reason why this option is true is because palmitic acid is actually a 16 carbon chain fatty acid. It's a saturated fatty acid, and upon complete oxidation, it yields close to 129 ATP, and this is definitely greater than what glucose can yield. So that's why the assertion is correct, the statement is correct, and definitely the reason why it yields more glucose is why it yields more ATP is because the number of carbons in palmitic acid are more reduced than those in glucose. So yes, the reason is also true for the assertion. So moving on to the next question. So when cell components are fractionated by sedimentation, the correct order from lower to higher gravitational force, that is G, in which the components get separated is what? So here clearly it says lower to higher, which means the organelles corresponding to bigger to smaller size. So if the organelle has a bigger size, then it requires lower gravitational force to settle it down. So bigger to smaller. So in this case, the correct option is answer A, that is nuclei, which is bigger in size compared to mitochondria, which is bigger in size compared to microsomes, which is again bigger in size compared to ribosomes. And hence, nuclei will require the least gravitational force to settle down compared to the others, followed by mitochondria, followed by microsomes, and then ribosomes. And hence, the option A is correct. So, moving on to the next question. In a population, the probability of a susceptible individual getting infected with SARS-CoV-2 is low when a majority of individuals in the population becomes immune to this virus. So this phenomenon is known as, so this is a very simple question. In fact, you would have heard a lot about this during the COVID-19 pandemic, and the correct option here is herd immunity. So this herd immunity protects the population which are not which haven't been exposed to the vaccine, say like a precaution. So that's why this phenomenon is called herd immunity, and this is very common, especially during any outbreaks like pandemics. So herd immunity protects a group of people that has not yet received precautions like vaccines. Okay, moving on to the next question. So given below are four reactions of the glycolytic pathway catalyzed by the enzymes E1, E2, E3 and E4 as indicated. Which of these enzymes is or are not part of the gluconeogenesis pathway? So here it is important to know that though gluconeogenesis and glycolysis seem a bit confusing, it is important to know which enzymes are common to both and which enzymes are not. So in this case, the correct options are A and D. So since this is just discussing the previous year's questions, I'm not going too much into the details. So here the option, that is the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate is catalyzed by E1, and if you look at this enzyme is part of glycolysis, but it is not part of gluconeogenesis. Similarly, conversion of phosphoenol pyruvate to pyruvate by pyruvate kinase is also part of glycolysis but not gluconeogenesis. However, the other two are part of gluconeogenesis as well. So that is why it is important to write down the pathway and then look at which enzymes are common to both and which enzymes differ in both the pathways. So the correct options here are A and B. So moving on to the next question. Which of the following molecules is or are second messengers produced by phosphoinositide signaling cascade? Here, if you look at the signaling pathway, which ultimately results in the production of calcium ions from the endoplasmic reticulum. The correct options here are B and D, that is inositol 1,4,5 triphosphate and diacylglycerol. So it is important to remember that it is 1,4,5 and not 1,3,5 because it can be easily confusing. And if you have this diagram, that is very common in most of the textbooks, where they would have mentioned which molecules ultimately get inside the cell through the endosomes and then catalyze these pathways, then it becomes easier to pick the correct option. So this is going to be inositol 1,4,5 triphosphate and diacylglycerol that ultimately results in the formation of calcium 2 plus ions release. Okay, and this is a very common question. Moving on to the next question. A protein has 7 cysteine residues, okay? The maximum number of disulfide bonds of different combinations that can possibly be formed by the 7 cysteine residues is in integers. The answer should be in integers. So first of all, a disulfide bonds would require 2 cysteine residues to form. So in that case, if I have the first cysteine residue taken, it can form disulfide bond with 6 remaining cysteines. So that is going to be 6 into. So once the first disulfide bond is formed, you will have remaining 5 cysteine residues. So if you take the third cysteine residue, then it can form disulfide bond with remaining 4 cysteine residues. So that will be into 4. Multiplied by, once that disulfide bond is formed, you have remaining 3 cysteine residues. So hence, the fifth cysteine residue can form disulfide bonds with 2 remaining residues. So that will be into 2. So ultimately, this is going to be 6 into 4 into 2, which is 24 into 2, 48. I hope this is pretty clear because this requires a different type of permutation and combination thinking and not like the normal ones. So you have to remember, first you take the 1 cysteine residue and hence it can form with 6. So once the first disulfide bond is formed, 2 cysteine residues are taken. So now you will have remaining 5. So out of the 5, the third cysteine residue, because 2 are already used up in one disulfide bond, the third one can form disulfide bond with the remaining 4. So you multiply into 4. So 4 combinations are possible. Similarly, for the last combination, 2 combinations are possible. So hence, if you multiply all of these, you get 48. I hope it is clear. So moving on to the next question. A lyophilized sample of 20 nanomoles of an oligonucleotide is dissolved in water and the volume of the solution is made up to 200 microliter. The concentration in micromolar of the oligonucleotide in this solution is in integer. So first of all, concentration in molarity is given by the formula number of moles divided by volume. Okay. So here the number of moles is 20 nanomoles, which is 20 into 10 power minus 9. And the volume here is 200 microliters. So you have to convert it into liter, which is 10 power minus 6 because it's in molarity. So this, if you calculate, you will get the answer to be 1 by 10 into 10 power minus 3 molar. However, they ask the answer in micromolar. So you will 1 by 10 into 10 cube into 10 power minus 6 molar. 10 power minus 6 molar is micromolar. So that will be 1000 by 10, which is 100 micromolar. So the final answer here is in integer is 100 micromolar. Okay, this is a very simple question. Any basic chemistry knowledge should help you to solve this question. Moving on to the next question. DNA in a 1 centimeter long chromatin contains 5 into 10 power 9 base pairs. The full compaction of this DNA within the chromatin is in integer. So first of all, here they have given that 1 centimeter long DNA has 5 into 10 power 9 base pairs. Okay. So if you take the classical B form of DNA, 3.4 nanometer will have 10 base pairs. If you remember the helix pitch basically. 1 turn. So 3.4 nanometer will contain 10 base pairs. So how much length corresponds to 5 into 10 power 9 base pairs? So that will be x is equal to 5 into 10 power 9 into 3.4 divided by 10. So that will give you an answer of 17 into 10 power 8 nanometer. So that will be 1.7 meter. Okay. So 1.7 meter is equal to 170 centimeter. So ideally 5 into 10 power 9 base pairs should occupy 170 centimeter if it follows this. However, it occupies only 1 centimeter, which means the fold compaction is given by the actual length that should be occupied by the DNA for the given number of base pairs. So actual length, which is basically 1 centimeter, divided by the observed length. So here it is 1 centimeter. So that will be 170 centimeter by 1 centimeter, and the final answer is 170. So hence the fold compaction here is 170. Instead of occupying 170 centimeter, it is occupying only 1 centimeter. So thus the fold compaction is 170. I hope this is a pretty simple question to solve if you know the basics. Okay, moving on to the next question. Okay, the intracellular, let me just zoom this in. Yes, the intracellular concentrations of ATP, ADP, and inorganic phosphate in 4 cell types are given below. Which one of these cell types has the most negative delta G for ATP hydrolysis? So first of all, you should know what ATP hydrolysis is. You need to write the reaction. So that is ATP giving ADP plus inorganic phosphate, right? Okay, so these two are the products and ATP is the reactant here. So just mark it. Okay. So we also know that delta G from basic thermodynamics concepts, delta G is delta G naught plus RT ln products by reactant concentration. Basically the reaction quotient. Okay, so products by reactant concentration. So we need to find the cell type for which the delta G is most negative. And for a given reaction, delta G naught is going to be the same because that is the standard free energy change. So this is not going to vary between different cell types because the reaction is the same. So now we will just write it as for cell type L. Let us start with that. For cell type L, delta G equal to delta G naught plus RT ln 5 into 1.8 divided by 3 because the products are ADP and inorganic phosphate. So if you simplify this, you will get delta G naught plus RT ln 3. Okay. Now for cell type K, delta G equal to delta G naught plus RT ln 3 into 1.3 divided by 3.9. So that will be delta G naught plus RT ln 1. Okay, so ln 1 is 0. Okay, moving on to the next cell type, that is cell type B. So for cell type B, delta G is delta G naught plus RT ln 2.7 into 0.7 divided by 2.7. So that will be delta G naught plus RT ln 0.7. Okay. And for cell type M, the last cell type, so that will be delta G equal to delta G naught plus RT ln. So what are the concentration? 8 into 0.9 by 7.2. It will be 8 into 0.9 divided by 7.2. So that will be delta G naught plus RT ln 1. So that will be 0. So now we know that for ATP hydrolysis, this is a very spontaneous reaction for which delta G naught is already negative. So if this term is going to be positive, then delta G is going to be less negative than delta G naught. Okay, if this term is negative, then delta G is going to be more negative than delta G naught. Okay, so when we look at all these options, RT ln 3 is a positive term, this is a negative term. Okay, and this is going to be delta G naught itself because this is 0. Okay, however, if you look at cell type B, this is negative and ln 0.7 is negative. So this is also negative. So delta G is going to be more negative than delta G naught. However, if you look at cell type M, this is going to be delta G is going to be delta G naught because ln 1 is again 0. So the cell type for which delta G is the most negative amongst all is cell type B. So the correct option here is answer C. Okay, so with basic thermodynamics formulae, you will be able to solve this simple question. Moving on to the next question. Which of the following amino acids has more than 2 acid-base groups? Clearly, from the given options, the correct answer is tyrosine. If you look at the side chain, it has this benzene ring with an OH group attached to it. So this OH being polar can become O minus And when it furnishes H plus, it acts as an acid, and when it accepts H plus, it acts as a base. However, if you look at the other three, these are clearly non-polar amino acids, and hence they don't act as acid-base groups. So tyrosine is the correct answer here. This is a very simple question to score. Moving on to the next question. So here, enzyme activity profiles as a function of time in the absence or presence of different types of feedback mechanisms are shown in the given figure below. So match the following feedback mechanisms with the corresponding profiles in the figure. So here you have four types, that is no feedback mechanism, and then negative feedback mechanism with short delay, and negative feedback mechanism with long delay, and positive feedback mechanism. So if you look at these figures, clearly you can say that for positive feedback mechanism, it is option 1, because here if you look, the enzyme activity keeps increasing and stays the same with increasing time, which means that even as the products are getting formed, the enzyme activity doesn't decrease. So basically there is no decrease in the enzyme activity upon product formation. So this clearly states that it's a positive feedback mechanism. And if you look at the negative feedback with no feedback mechanism, basically this first option, it is going to be option 4. So here P corresponds to option 4. This is because the enzyme activity increases and then decreases. So basically this could be due to consumption of the enzyme and is not based on the product. However, if you look at the other two graphs, that is enzyme activity corresponding to time, and if you look at the signal, there is going to be there is a negative feedback mechanism here. So here as the product gets formed, the enzyme activity decreases. And once the enzyme activity decreases, the product also decreases. And once the product decreases, now the enzyme activity increases. So like there is a negative feedback here. However, you should choose between which one has a long delay and which one has a short delay. So the 1, 3 here is basically the one with short delay, and the one here 2 is basically the one with the long delay. So this one is the long delay, this one is the short delay. So yes, and hence the correct option here is option D, that is P matches with 4, Q matches with 3, R matches with 2, and S matches with 1. So that is the correct option, and this is how you deduce how feedback mechanisms can be correlated with the enzyme activity. Okay, moving on to the next question. So a linear fragment of 5 kilo base pairs, when completely digested with ECO R1, produces 2.5 kb, 1.5 kb, and 1 kb fragments. Complete digestion of the same 5 kb fragment with XBA1 produces 3.5 kb and 1.5 kb fragments. Which of the following sets of fragments will be obtained if this 5 kb fragment is fully digested with both the restriction enzymes simultaneously? So basically, you have for ECOR1, you have 2 restriction sites that will give you 2.5, 1.5, and 1. And for XBA1, you have 1 restriction site that gives you 3.5 and 1.5. So basically, what you have to do is here, it has to be through trial and error, or if you're lucky enough or smart enough, you can get it in the first go. So here you need to arrange this 2.5, 1.5 and 1 in different orientations, and then XBA1 in different orientations. So this will have like 6 possibilities, and this will have like 2 possibilities, because if 2.5 is first, then you have 1.51 or 1.5, and similarly if you have 1.5 as the first first fragment, you can have 2.5 here and 1, or 1 and 2.5 here. So you will totally have like 6 possibilities here. And for XBA1, it can be either 3.5 1.5 or 1.5 3.5. So now if you introduce both the recognition sites in the linear DNA fragment and do a double digestion, ultimately you end up with the answer as option number C, wherein the ultimate the restriction map would be like this. That is, you have an ECOR1 site here, you have an XBA1 site here, and then you have an ECOR1 site here. But here it will be 1.5, 2.5, 1. So here Eco R1 gives you 1.5, 2.5 and 1. So that is matching. Yes, correct. And XBA1 gives you 3.5. Yes. And then 1.5. So this will give you, so if you basically introduce both the the recognition sites for both the restriction enzymes and then look at your fragments and the options, this is the correct answer. So you just have to do this trial and error method and figure out which one is matching and which one also matches with the given options, and you don't have to worry about the other options also being correct. This is the correct option. Okay, so that's how you do it. Yeah, okay. Moving on to the next question. Yes. So this one is pretty simple. Match the cell types listed in group 1 with the associated processes listed in group 2. Of course, if you know basic immunology, this is going to be a very easy peasy question. So here NK cells, they have antibody dependent cell mediated cytotoxicity. They are natural killer cells and they do that. And B cells, anybody would know it is involved in antibody production. Mast cells, yes, histamine production, which are involved in inflammatory pathways, and neutrophils are the first cells to be recruited at the site of infection. So this is the matching pattern, and the correct option here is C. Yes, so this is a very simple and easy question to score full marks. Moving on to the next question. Yes. 4 statements about lipids are given as below options. Choose the statements which is or are correct. So this is a multi-select question, so you have to be really careful with the options. So here the first statement is cholesterol is amphipathic. Absolutely true because it has an OH group. And the second option is self-assembly of phospholipids in water is due to hydrophobic effect. Absolutely true because it's a repelling environment for the phospholipids. They are hydrophobic in nature predominantly. And the third one is the temperature at which the gel phase changes to liquid crystalline phase increases with an increase in the degree of unsaturation of fatty acyl tail. So this is not true. It's the other way around. And the choline head group of lipids is positively charged. If you look at the structure of, say, phosphatidylcholine or basically the choline head group of these structures in your basic biochemistry textbook, it has a positive charge group. So this option is also correct. So the correct options are A, B, and D. Moving on to the next question. So which of the following techniques can be used to separate proteins according to their molecular weights from a mixture of proteins? So here it's a very simple question, and we have all done most of these techniques during our practical classes, and the correct options are B, yes, size exclusion chromatography, because it's also called the gel filtration chromatography. The large ones elute first, followed by the smaller ones. And then SDS page, yes, sodium dodecyl sulfate polyacrylamide gel electrophoresis. So in SDS page, the proteins get separated. The migration of proteins basically depends on their molecular weight, and of course sucrose density gradient centrifugation based on the size and the shape. A different gradient of separation of proteins is observed in this centrifugation method. Okay, ion exchange, that's the wrong option. Yes. Moving on to the next question. So B cells produce two forms of immunoglobulin, which is membrane-bound form known as the B cell receptor, and two, the soluble form known as the antibody. Yes. Which of the following statements is or are correct about BCR, an antibody produced by the same B cell? So here, first of all, you should know how antibodies are produced and basically the arrangements of the genes that is involved in producing these. So the first option is BCR and antibody have identical antigen binding site. Yes, that is true. BCR and antibody recognize different epitopes. No, since they are from the same genes and they have the same variable regions as well. BCR and antibody are encoded by the same gene. Yes, that is also true. And BCR and antibody are formed by differential splicing or the alternative splicing. So if the membrane bound region of the gene is also included, then it is going to be the BCR, or if not, it's going to be antibody and is going to be secreted when the B cell recognizes the appropriate antigen. So the correct options here are A, C, and D. Okay. Moving on to the next question. A 100 ml solution of pH 10 was well mixed with a 100 ml solution of pH 4. The pH of the resultant 200 ml solution is, so this is a very basic chemistry question. So first of all, you have two solutions, that is 100 ml of pH 10 and you have 100 ml of pH 4. Okay. So here the concentration of, pH is equal to formula minus log of H plus concentration ion. So here concentration of H plus is equal to 10 power minus 10 molar. And here concentration of H plus is equal to 10 power minus 4 molar. Now you need to know the number of moles here. So number of moles is equal to 10 power minus 10 into 100 into 10 power minus 3, because this is 100 ml and it's in molar. Let's also have the volume in liters. So I multiply by 10 power minus 3. And here it is 10 power minus 4 into 100 into 10 power minus 3. So here for a 200 ml solution, the total number of moles is going to be this plus this. So that is 10 power minus 10 into 100 into 10 power minus 3 plus 10 power minus 4 into 100 into 10 power minus 3, and now the total volume is 200. So 200 into 10 power minus 3 liters. 10 power minus 3, 10 power minus 3, 10 power minus 3, they get cancelled, and 100, 100, 200 will become 1 by 2 here. So 1 by 2 into 10 power minus 10 plus 10 power minus 4. So 10 power minus 10 is way less than 10 power minus 4. So I'm just going to ignore the term. So that will be equal to 1 by 2 into roughly. So I wouldn't say equal to, approximately into 10 power minus 4. So now this is the concentration of H plus. However, you need to find the pH. pH is equal to minus log of 1 by 2 into 10 power minus 4. So if you know the basic logarithmic rules, that is log of A into B is log A plus log B. So that will be minus log of 1 by 2 plus minus log of 10 power minus 4. So this will be 2 if you know the, log 2, which if you know the log of 1 by A is minus log A, already there is a negative sign, so it's going to be positive, plus log of 10 minus log of 10 power minus 4 is going to be 4. So 4 plus log 2. Log 2 is approximately 0.3, and if you add it, you get 4.3. So the pH of the solution, 200 ml solution, is going to be approximately 4.3. Okay? So this is a very simple question to solve. Moving on to the next question. Yes, this is a really good question that I would like everybody to pay quite good attention to. So here, an organism uses only the glycerophosphate shunt pathway to transport cytosolic NADH to mitochondria. For every 2 electrons transported, complex 1, complex 3 and complex 4 of the electron transport chain in this organism transport 2.5, 1.5 and 2 protons respectively. The H plus to ATP ratio of F0, F1 ATPase of this organism is 4. Terminal electron acceptor is oxygen. The number of ATP molecules synthesized by oxidizing NADH from glycolysis is what? You have to round it off to 2 decimal places. Okay. So here, first of all, you should know that from glycolysis, you get 2 NADH, right? 2 NADH. If you know the pathway, I'm not going too much into details because I'm only discussing the previous year's questions. And this NADH can get into the mitochondria via 2 pathways, and one such pathway is the glycerol 3-phosphate shuttle or the glycerophosphate shuttle, as they mentioned in the question. So if it goes through this particular pathway, it enters the mitochondria as 2-FADH2, and upon oxidation, it gets converted to, this FADH2 gets converted to ATP. It helps in the formation of ATP basically. Okay, so when FADH2 is involved, you have to consider only the protons of complex 3 and complex 4. If you look at the mitochondrial complexes, how H plus ions are transported. So you have to only consider 3 and 4 if FADH2 is involved. So that is something, that's that theory which you need to know. So for every 2 electron, and this is for FADH2 I mean, you have for this particular organism 1.5 plus 2, which is 3.5 hydrogen ions being transported, right? Yes. However, this is for only 1 FADH2. For 2 FADH2, this is going to be 2 into 3.5, which is 7 H plus, right? Yes. And now they have also given in the question that the H plus to ATP ratio is equal to 4. What do you mean by that? It means that you require 4H plus to produce 1 ATP. However, we have 7H plus here. So for 7H plus, what's the amount of ATP that you need to produce? So that will be equal to 7 by 4, that is 1.75. So via this shunt, the number of ATP molecules synthesized by oxidizing NADH from glycolysis is 1.75. I hope this question is pretty clear. Moving on to the last question. Here, if the extracellular concentration of sodium ion is 10 times more than its intracellular concentration, then the sodium equilibrium potential at 20 degrees Celsius in millivolts is what? Assume that the membrane is only permeable to Na plus ions. Okay, so here we use what is called the Nernst equation, which says that NF E cell is equal to RT ln K. So here E cell is the equilibrium potential, F is the Faraday's constant, N is the number of electrons, R is the universal gas constant, T is the temperature, and K is the equilibrium constant. So here we need to find E cell. So E cell here is RT by NF into ln K. Okay. So here I am using the standard constants, because when I tried solving this using the constant that they gave, the answer key doesn't match. So I will try doing it using the standard constants for these. So taking R as 8.314 and temperature in Kelvin. So here temperature is given as 20 degrees Celsius. So for converting to Kelvin, you will add 20 plus 273.15. So that will be 293.15, and Faraday's constant, I take it as 96,485, and here N is equal to 1 because it's sodium ion, so the charge is 1, and here K is equal to concentration of sodium ion in the so K is equal to concentration of sodium ion in the extracellular membrane, in the extracellular space, sorry, not membrane, extracellular space, divided by concentration of sodium ion in the inside of the cell. Okay, so that will be equal to 10 by 1. Okay, so now substituting all of these, you get E cell is equal to 8.314 into 293.15 into ln 10 divided by 1 whole divided by 96,485. If you solve this, you will ultimately get the answer to be, this is in volts, so make sure you change it to millivolts. So you will get the answer to be 58.2 millivolts, that is mentioned in their answer key. So by taking these constants, you get the answer to be in this range. However, I hope this discrepancy won't be there in the coming papers. Anyways, so that's how you solve. And with this, I'm completing my discussion for the answer key for the previous year's questions of Biochemistry GATE Excel 2023 paper. I hope you found this video useful and hope you ace the upcoming GATE examinations. So stay tuned for more answer key discussions. Hope you have a nice day. Goodbye.